Okay, so we've been thinking a lot about the rules that we have from meiosis and fertilization and how they can be applied to different phenomena. We looked at Mendel's pea plants, we looked at mice, and we looked at chickens. And the chickens kind of broke our rules a little bit. Specifically, they broke our rule of dominance, and they were an example of codominance. We're going to look at ball pythons to see if they follow our rules or if they break our rules too. Some ball pythons are brown and some are white. We call that leucistic. Let's check our rules to explain this trait. It also gives us a chance to review what was going on all the way back at the beginning of our genetics unit, where we think about, do we think the DNA is different in the white snake from this brown snake? And what does that mean for the protein? I'd like you to have open in front of you Big Ideas, page 17. And I'd like you to work off of 3.4, Do Our Rules Explain Ball Pythons in your activities packet. So on the top of that 3.4, Do Our Rules Explain Ball Pythons, you're going to see what I hope is a very familiar chart, which has our DNA, RNA, amino acids, and proteins. Use this to compare the DNA of the brown python and the white python. Complete this table and pause me and check in when you're done. Okay, so hopefully you were able to convert your DNA into RNA and your RNA into amino acids, and you can use this screen to double check your answers. And I hope that you noticed that on this last amino acid for a brown python, that was very different from the last amino acid from the white python. This one letter difference meant a different RNA, which led to a different amino acid. Now, different amino acids can lead to different shapes of proteins, and that can lead to different traits. So now that we've had a chance to review what's happening at the molecular level with different traits, let's go ahead and look at our gene pathway. I hope this looks familiar to you. Ball pythons function very similarly to humans, where we have a colorless molecule. And when you have a functional tyrosinase protein, it can be converted into a colored melanin pigment molecule. So how does this pathway change in a white snake? Well, think about what happens if this tyrosinase protein is no longer fully functional because it has a different amino acid. Let's take a look using our cell models. Again, I hope these cell models look super familiar to you, where we have two functional alleles for our brown snake and two non-functional alleles for our white snake. Predict what will happen in these cell model animations. I hope you predicted that with two functional alleles, our white molecules will move through our fully functional tyrosinase enzymes and get converted into melanin molecules. But when we have non-functional alleles, that means we have non-functional tyrosinase enzymes. That means those molecules are just going to stay white. So this model of a brown snake and this model of a white snake looks an awful lot like what's going on with human albinos. So what do you think is the dominant trait? Jot down on a piece of paper what you think is dominant. Brown or white? How would we figure that out? Well, first of all, we're going to cross them. Whatever the dominant trait is, should be what we see in the offspring. So if brown is dominant, we would expect to see brown offspring. If white's dominant, we would expect to see white offspring. That's assuming it follows our rules. So let's check it out. Here's our moment of truth. The babies of this brown snake and this white snake are, whoa, they are light brown. They aren't dark brown, they aren't white, they're a lighter brown. Well, that kind of breaks our rules. So And this isn't like the way the chickens broke our rules, because the chickens were part white, part red. They were half and half. That's not what happened here. So we have discovered something new. And you can jot this down on Big Ideas, page 17. We call this incomplete dominance. This is when our hybrids or our heterozygotes show a different trait. That's often the blend of the trait. Neither trait is going to show completely. So in this case, it's not brown, it's not dark brown, it's not white, but it's light brown, somewhere in between. So you can write that down in your big ideas. So if we come back to the way we might model this particular cross that we've done, you could use that blank sheet on the back of your big ideas packet or a blank sheet of paper to just model this cross. Now I want to remind you that the letters that we put down to represent our crosses and models are just letters. They're variables. You can really pick whatever you want. In fact, some people might use apostrophes, big B and big B apostrophes. Some people might like to keep using the capital letter and lowercase letter. In this example, I'm going to use a capital B to represent the brown allele and a capital W to represent the white allele. All right. So if we know that our white female parent is a big W, and our male parent is a big B, we know all that she can make in terms of eggs are a big W and a big W. And all this male parent can make in terms of sperm are a big B and a big B. And when they come together, the big W and the big W come together with the big B and big B, all we can get are big B, big W's. So that means big B, big W, that's what our light brown snake is. That's its genotype. All of these are going to look the same. All of them are going to look light brown. All right, so if we went ahead and thought about what that looks like in these generations, we're going to get 100% that are that big B, big W. But the question about why are they light brown instead of being spotted or instead of just showing one trait, that's really interesting. What could cause that to happen? Well, let's try another cross, big B, big W, and see what we think will happen there. Make an egg and sperm chart, make a Punnett square, and see what you think will happen with their offspring. Pause this and come back to me when you're done. All right, so if they're light brown, big B, big W, this parent can make a big B egg and a big W egg. This parent can make a big B sperm and a big W sperm. In your Punnett square, again, these are the parents. They can make big B, big B, big BW, or big W, little w. These are all the different combinations of the eggs and the sperm. What percentages and ratios can we get from this, knowing that these are our combinations here? And again, you can think about this. Well, all right, our brown snakes were the big B. They made up one of those four, so that was 25%. Our light brown snakes, those are the big B, big W, we had two of those, two of the four. That's 50%. Our albino or our white snakes would be 25%. One of the four. Our ratio is a 1 to 2 to 1. So, were our predictions correct? Let's go ahead and see. Look at this. When we cross those two, we did end up getting one brown, two light brown, and one white offspring. Now, I imagine if these snakes had lots more babies, that we could see slightly different ratios. They aren't going to be perfect every time. Each time they have a new baby, they will have a 1/4 of a chance of having a white snake, a 1/4 of a chance having a darker brown snake, and a 50% or half a chance of having this lighter brown color. We still haven't really answered the question, though, about why incomplete dominance happens. And is this light brown snake actually producing a functional protein? And how do we know? This is an amazing question that our model, our cell model we've been working on all the way back from the beginning of this unit can help us answer. Remember, in order to get a light brown snake, we need to be getting some of this melanin pigment molecule. We've got a colorless molecule here. But what we notice, it's a light brown snake. So what does that mean? How could we use our model to explain we're not getting quite as much melanin as normal? Well, let's go ahead and take a look. Again, two functional alleles have plenty of functional tyrosinase, and they're making plenty of melanin. But somehow, in this case, we are making less melanin. Excuse me. So predict what will happen when you cross a brown snake and a light brown snake. Go ahead and make an egg and a sperm chart or a Punnett square to make your predictions. So your predictions probably looked something like this, where you can have a big B and a big W, a big B and a big B. They can only make big B sperm. They can make a big B or a big W sperm. And that they make these different combinations. And when you count them up, you see, wow, 2 of the 4 offspring are going to be brown, and 2 of the 4 offspring combinations are going to be light brown. So in that case, we would have a 50% chance of having a brown snake, a 50% chance of having a light brown snake, 0% chance of having the white snake, a 1 to 1 to 0 ratio. So were our predictions correct? Let's go ahead and cross our snakes. And once again, we are at about half and half. Yes, half of them are light brown and half of them are dark brown. Oh, but you know what? If they had another one, it's still a 50% chance. And so it might not end up being even. It might end up being perfect every time. How could we explain a snake like this? What would we call this? You might be using the word codominance, because some cells are making the pigment and some cells are not. How can cells that have the same DNA have different proteins? We've talked about this before. This reminds us of our chickens that have red and white spots. We've also asked this question before. If every single cell has the same DNA, what could cause it to have different proteins? Think to yourself, what have we used to explain something like this before? I think we've talked about genes being turned on and genes being turned off. So in this situation, in this cell, the tyrosinase gene is turned on, which means the tyrosinase enzymes are being made and they can make the white molecule turn into a brown melanin pigment. In these cells, the tyrosinase gene is turned off. That means it is not even making any of the tyrosinase enzyme. It's not that it's not functional, it's just not making it at all. What would cause a pathway like this to sometimes make the tyrosinase protein and sometimes not? Pause for a minute and think about that. I'm hoping you came up with another regulator gene. Again, this is review from our very first unit. One gene can turn another gene on and off. So if we had a functional regulator gene turning it on, we're going to make plenty of this. If we have a non-functional regulator gene, it's going to turn it off and say, no, I'm not going to make that molecule at all. So in review, what do hybrids look like? If it's normal dominance that follows our rules, what would the hybrid or the heterozygote look like? If it's incomplete dominant, what would the hybrid or heterozygote look like? And if it's co-dominant, what would the hybrid or a heterozygote look like? Make your predictions, pause me to check them. All right, if it's standard dominance, we are going to see one of the two traits showing up. We would expect to see one or the other. So in this case, red is dominant. If it's incomplete dominant, we would expect to see somewhere in between. We are making some of that, so we might get a pink between a red and a white flower. We're making some of the protein. If it's co-dominant, we would expect to see some of each, which means in some cells, the gene is turned on, getting the red coloring, and in some cells, the gene is turned off, giving the white coloring. I hope this was a nice way to bring together all of the various ideas that we have taught in this unit. Please remember to email us with questions.